All right, so we are on slide 18 of the chapter 25 PowerPoint lecture. We were looking at the structure of HIV. There are two glycoproteins on the virus, on the envelope of the virus. They're called GP41 and GP120. Those bind to, on the white blood cell, which are T cells, they bind to a receptor that's on the T cell called CD4. And that is the first step needed to trigger endocytosis of the virus particle, and that brings the virus into the white blood cell. So this is one of, remember, 3 ways that viruses can get into an animal cell, and ultimately the viral chromosome and the reverse transcriptase will be released into the white blood cell. So this picture shows the same process, but it goes even further. So you see the attachment of the GP120 and GP41 to the CD4, which is on the host cell. It brings in the virus. The virus is uncoated, and the reverse transcriptase and the RNA chromosomes are released. The reverse transcriptase copies the RNA into DNA, which then goes into the nucleus and inserts into the host cell, the white blood cell's own chromosome. So that virus DNA embedded in the host cell chromosome is called the provirus. You may recall that when a phage chromosome integrates into a bacterial chromosome, that's called the prophage. But with an animal virus, it's called a provirus. But it's still a similar function. The viral chromosome is going to be latent. It's going to be dormant until such a time as it becomes activated. There are things that can cause activation. Sometimes another illness can activate the HIV virus within the white blood cell. Hormones can sometimes cause that, but you can have a dormancy that can go for years with HIV. And once it becomes activated, though, the genes on the provirus, on the viral chromosome, will be expressed, just like the genes on the on the human chromosome. That produces mRNA, which is then sent out to the cytoplasm and is translated by a ribosome. All of this is the host cell machinery. What it produces, new virus particles, which then are budded from the membrane to make a new envelope. And that process requires a protease enzyme, and so that's why protease inhibitors have been effective as a treatment for HIV, because it blocks and inhibits the protease, which helps new particles bud from the membrane. But that's the normal replication process of HIV, and this can go on and on until the white blood cell is just totally drained of ability to continue, and they die, and your white blood cell counts go very small. And this is maybe an overly complex graph of the course of AIDS, each line on the graph tracking a different aspect of the progress of the virus and the disease. So the red line shows how much HIV is in your blood. So when you start off, you get initially kind of a peak after a couple weeks, and then it goes down, and the HIV is going into your cells and becoming dormant. You can see on the green line, your T cells are just decreasing over time, and you have kind of an increase and then a decrease of antibodies against HIV. But ultimately, what's important here is that the very end point of the disease. when you get towards, and this is just a kind of a general graph, let's say you're at year 8, you've had a latency of the virus for a long time, but once the virus is reactivated, it looks like maybe in this graph around year 7 to 8, then you notice that the amount of HIV in the blood increases rapidly, the amount of T cells decreases rapidly, and you ultimately succumb to some other disease, some other pathogen, and you die basically from that, although the HIV is the underlying cause of your immunosuppression. We will look in the next, after this chapter, we'll be looking at the World Health Organization data on HIV and look at where we're at in 2018. Your book tends to stop its data with 2015 typically, so I'm gonna show you where to look on the World Health Organization to find, up-to-date information on the distribution and the incidence of AIDS and HIV in the world. All right, so we switched gears and we are looking now at respiratory syncytial virus, which is RSV. It's a very dangerous respiratory disease for babies, and it's because the virus actually causes the lung cells to fuse. The fusion of cells creates larger cells that have multiple nuclei, and those types of structures are called syncytia. And so a syncytia is a many cells that are fused together, or a large cell that has many nuclei as a result. And so it basically reduces your surface area of your lung tissue, and you may recall from anatomy or another course that surface area is important for materials to pass, in this case oxygen, to get into your blood. So it becomes a problem if you have less surface area. In an adult or even older child, it's going to be less of a problem and often asymptomatic or like a mild cold, but it turns out that your immune system will actually increase the amount of damage to your lung tissue, again, mostly for babies. This picture just shows multiple cells fusing together. The nucleus is shown as sort of this pink structure here, and once the cells are fused together, of course new viruses are being produced as well that are budding from the syncytium and invading the nearby cells. All right, our last disease in this chapter is rabies, and rabies is another of the zoonotic group, meaning it's passed from animals to humans. Its main effect is on the central nervous system, and it's irreversible and doesn't matter if you're a healthy person or an immunocompromised person. Anybody can be infected because it's a very effective virus. And so there's no such thing as asymptomatic rabies. You will get spinal cord and brain degeneration. Your brain and your spinal cord, your nervous tissue turns to mush. And some of the manifestations are going to be seizures, disorientation, hallucination, paralysis, and then I highlighted hydrophobia because that's kind of an unusual but very characteristic of rabies. It must affect some part of the brain that involves your opinion of water, because you become absolutely terrified of water. So that's called hydrophobia. And that's very characteristic of rabies. So an animal with rabies will be terrified of water, and a human as well. Death results from respiratory paralysis, and basically your brain stops controlling everything that it needs to do. So like I said, it's a zoonotic disease, meaning it's passed from animals to humans. The primary reservoir in the city would be dogs, although bats a bite of a bat is often how you get it. You can get it from a bite of a dog as well. If you get bit by a dog or a bat and they can't find the animal, then they'll probably start treating you for rabies, because if you don't have it, there's no harm. But if you do have it, you must get treatment before symptoms start to appear. Once symptoms start to appear, it's too late to do treatment. And they've done autopsies on individuals who die of rabies, and there's these formations, these staining inclusions that occur in the brain tissue called Negri bodies. And that's a very diagnostic, of course, after the fact, post-mortem diagnostic of the fact that it definitely was rabies. Over here on the left, you see the rabies virus. I think it's kind of interesting that it has a bullet shape, because that's basically, I mean, it's a very lethal virus, so I think it's kind of fitting that the shape of the virus is like a bullet. Oh, I forgot. Influenza is the last one. Influenza. Influenza is interesting because it has a segmented genome. It has 8 single-stranded RNA molecules in each virion, or viral particle. The important part of influenza, or at least the part that you may have heard of, has to do with 2 of its glycoproteins. Now, its glycoproteins are encoded by its genes, and you have what's called neuraminidase, that's a surface glycoprotein, and you have hemagglutinin, which is another surface glycoprotein. The neuraminidase is also an enzyme, but it's a glycoprotein enzyme. And so you've probably heard of H1N1. Well, the H is hemagglutinin and the N is neuraminidase. And there's like H1N1, but there's also like H3N2 and H2N4 and all such. So different strains, slightly different mutant versions of each of these create slightly different serotypes of the flu. And that's why every year a different combination of flu comes in. There's mutations in the genes themselves that encode the neuraminidase and the hemagglutinin, but then also you can have a new combination of these mutants when one cell gets infected with 2 different flus, which can happen. And so this is called antigenic drift and antigenic shift for the flu. Antigenic drift is new mutations when the chromosome is being replicated within a infected cell. Some of the chromosomes might incorporate a new mutation, and so that actually, when that new virus buds off of that host cell, it carries a slightly different strain, a newly formed strain from a mutation. That's called antigenic drift. Antigenic shift is when 2 different strains of flu co-infect one cell, and different combinations of those 8 segments end up in a new virus particle. And it can be different than the original 2. And really, to make things more complicated, you could actually have co-infection of one cell with 2 different strains, and then on top of that you could have new mutations happening during replication. So this could all actually happen simultaneously. You could have antigenic drift and antigenic shift, and that would really create some new viruses. So the thing is that every year as the virus is going around the world, both of these are happening, the drift and the shift. And so you're virtually guaranteed to get some new combination or some new slightly different virus form. And that's why they have to keep coming up with new vaccines. Now, I would say that when you get the vaccine, even if it's only 50% effective, that's 50% more than what you had at the beginning of the season, but you do have some protection from previous strains, because some segments of the flu come back every year. So over time, as you get more and more immunizations, it should ultimately build up. But if there is a weird mutation that comes around every once in a while, then yes, you could get sick. But you should get your vaccination every year because it adds up over time to really protect you. And we talked about the herd. You really want to protect the herd, not just yourself. So the symptoms of flu are fever. Malaise just means you feel exhausted, tired, you don't want to get out of bed. Headache and myalgia. Myalgia is muscle pain, so the aches, you get the muscle aches. The influenza virus comes in through your respiratory system, so somebody else coughing and sneezing and such spreads it for the most part. It incubates a day, so I don't know about you, but there have been times when I felt fine in the morning, and by the end of the day I was starting to feel pretty bad, because you remember how fast virus can replicate. It kills your epithelial cells in your lungs, so it really causes long-term damage. So as much as you can, you want to avoid the flu. And that is the end of our Chapter 25 lecture. If you have any questions, be sure to send me emails. I will be responding to my email and my remind over the break.